Good morning to everybody. Welcome to A Coruña. I am Marga Poza from the Microbiology Research Group of the Biomedical Research Institute of A Coruña. It's a great pleasure for me to open this workshop, and on behalf of the organizing team, we wish to thank you for coming here to A Coruña to this workshop. Well, if you need any information, do not hesitate to ask any member of the organizing team. We're wearing all the orange stickers, as you can see. So we are here for anything. And what is hard? I hope those persons who came yesterday to the guided tour through the A Coruña City Center enjoy it too much. And you have to know that today we have a networking dinner at the Queen's Tavern in Malepita. So if you want to come join with us, you have to sign in the registration desk that's there as soon as possible, please.
Hi, I'm Joaquin Vierna from AllGenetics, and I'd like to thank the speakers on behalf of everyone here for accepting the invitation to join us for the workshop. We're very happy to have you all at the FICA and are very impressed, or quite impressed, by the number of people interested in learning about DNA metabarcoding. And thank you very much also to our sponsors, who are the Consorcio de Turismo y Congresos de A Coruña and Celta Ingenieros. And I think that's it. We can start.
We can start with the first session. The first speaker is Antón Vizcaíno. He's going to talk about intro to the DNA metabarcoding from experimental design to sequencing. Antón studied biology at the University of A Coruña and Santiago de Compostela. He has been working in AllGenetics since 2012, and since 2014 he is AllGenetics head of laboratory and partner. He has worked on hundreds of genomics and transcription projects, most of them using the Illumina sequencing technologies, and he's currently doing his PhD on third-generation sequencing technologies, focusing on technical aspects of Oxford nanopore sequencers.
Good morning, everyone, and I would also like to thank you all for coming on behalf of the organizing committee. And so to open this workshop, I have prepared this talk that I have titled Intro to DNA Metabarcoding, from experimental design to sequencing. And I will try to summarize the main important aspects of this technique. I know that many of you have been using this technique for quite a long time now, but I'm also aware that some of you might be new to it. So therefore, as a disclaimer, I have to say that I'm going to try to keep this talk as basic as possible, and it is just intended to make sure that we're all on the same page, and that we are all starting from some common ground in order to get this workshop started. So, as I have said, I will try to cover all of the main stages of a typical DNA metabarcoding workflow. And so first of all, we have to talk about what DNA metabarcoding is. So DNA metabarcoding is a fairly recent molecular biology technique that relies on the power of high-throughput sequencing in order to recover all of the biodiversity present in a complex sample. And when we talk about a complex sample, we mean, in this context, we mean a sample that is made up of specimens that are hard or impossible to individualize. So they have to be studied as a whole. And so, we start with that complex sample. As I said, we extract the DNA, we sequence it, and then we're able to tell which species that sample was made up of. And just to get a little bit more into detail, this is the typical DNA metabarcoding workflow. And so we start with that complex sample, as I had said, and this sample can come from a variety of origins, such as soil, water, or fecal matter, or even herbal blends, or hamburger meat, and we want to know what kind of different species made up that hamburger. And so yeah, we start with that complex sample. Then we have to extract the DNA as a whole, meaning that we can't individualize the specimens here. So we're not separating the specimens before we extract the DNA, but we extract the DNA as a whole. And then with that DNA, we prepare what we call a library, which is a set of amplicons. So we're amplifying the DNA barcode, which is a taxonomically relevant region in the genome that varies between different taxonomic groups. And what we're doing is we want to prepare those molecules for sequencing. And we do that by adding some oligonucleotide tails at the molecule's ends in order to make it ready for sequencing. What we do then is we sequence those samples, and we obtain millions, ideally millions of sequences that are going to be then bioinformatically analyzed and assign their correct taxonomic identity. So to get us started, I would like to go through the main points that we need to consider when performing one of these experiments. So the first thing we need to be thinking about is the experimental design, how we're going to perform our experiments. And the first thing we need to think of are the goals of our experiments, and to see if DNA metabarcoding is the technique that we need, or maybe not. But once we have decided that it is, then we have to start thinking about the number of samples that we need and the number of replicates. Of course, the more the better usually. But of course, we need to find a balance between that and the economic issues, of course, because this doesn't come for free. And then we need to be thinking about which taxonomic groups we want to target. So unfortunately, there are no universal primers to target all of the world's biodiversity. So we need to stick to some taxonomic groups that we might be interested in, and design and select our primers accordingly. And one other thing we need to keep in mind is the expected biodiversity of our samples in order to know how much we need to sequence. We're going to need to sequence more if we are dealing with a more complex sample than if we're dealing with only a few species. So, for example, if we're targeting bacterial groups, we're going to need more sequencing than if we're targeting, say, invertebrates or something more, less diverse. So once we have our experimental design ready, we can go on to sample collection. And there's a few things that I'd like to comment on this. First of all, we need to avoid cross-contamination at all costs, because since high-throughput sequencing technologies are so powerful, if we have any contamination, it's going to show, and we're probably not going to be able to detect it. And so this is a major point I want to make. Avoid cross-contamination in the field by sterilizing all your material before you go from one sample to the other, making sure that there is no sample-to-sample carryover, and making sure that you're doing everything right. Also, another thing we need to be thinking about is how we're going to preserve our samples from the field, in the transport from the field to the lab. And what we're seeing here is that the best way to go, at least for some types of samples, is to just use the preservation buffer that comes with usually the DNA isolation kits. That's what's been working for us. And it's also important to keep in mind the temperature and to keep our samples chilly in order for the DNA not to get degraded. There are some. So once we have our sample, and once we have taken them back to the lab, we want to extract our DNA as soon as possible. And the first thing we need to be thinking about is which DNA isolation kit we want to use, and that's going to be dependent on the type of sample that we have. So there are kits available in the market for soil DNA extraction, for water DNA extraction, for fecal matter, for highly degraded DNA, etc. So we need to choose the DNA isolation kit that works best for us, or even preparing our own buffers. Then it's important to at least perform one extraction blank per round of sequencing, just to make sure that we're not contaminating our samples during this process. And once we have our DNA isolated, we're going to need to check the DNA concentration and the DNA integrity, as well as any inhibitors that might be problematic in the downstream processes. So once we have done all of that, once we've made sure that we have enough DNA and of enough quality, we can go on to library preparation. And library preparation is just to prepare the DNA for sequencing. So there are several ways to do this. We usually follow a 2-PCR approach, which is the one I'm going to show here, but there's going to be people later talking about different ways of making the libraries. So I'm just going to stick to this one for the moment. But you will see that there are more ways to do this. So as I said, we are following a 2-PCR approach. So PCR1, and then we'll talk about PCR2. For PCR1, what we want to do is we're going to amplify the DNA barcode I've talked about before. And the DNA barcode is going to be different for each taxonomic group that we want to target. So for example, for bacteria, it usually is the 16S ribosomal RNA gene. For fungi, it might be the ITS gene. For invertebrates, the CO1, for example. So we want to design our forward and reverse primers in order to amplify that region we're interested in. And this is done by designing the primers and attaching another sequence to the 5' end in order to make the molecule ready for sequencing. And so after several rounds of PCR, what we end up with is this construct right here with the forward primer binding site, the reverse primer binding site, and something called the sequencing primer, which is going to be needed for sequencing later. So once we have this, we're going to use this as the input for our PCR2, which so this is the input. That's the product from PCR1 that we got, and PCR2 is made pretty much in the same way, so following the same idea, and but this time our primers are going to be more universal. So they're not going to be taxon specific anymore. They're going to be specific to the sequencing primer, which is the artificial sequence that we just added in PCR1. So we can use these primers for all of our DNA metabarcoding projects or experiments. So these primers are also fusion primers that consist of 3 different parts. The sequencing primer, as I have said, it's useful for sequencing. Also, the adapter is the molecule that is needed in order for the molecules to attach to the flow cell of the sequencer. So these 2 sequences are needed for sequencing, and then we have the forward index and the reverse index. The indices are 8 base pair long oligonucleotides that are different for each of the samples that are to be analyzed at once. Therefore, each of our samples is going to have a different forward and different reverse index. So what we can do with this is we can pool several samples together and sequence them all at once. And then by looking, once we get the sequences from the sequencer, we can look at this part right here of our construct, here and here, and know which sample the sequences have originated from. So we're able to tell which sample this rays come from. So once we have our libraries ready, as I said, we're going to pool them all together, and in order to load them into the sequencer. So here we have 3 samples. Each sample has different molecules. So these are different amplicons ready for sequencing. Each of these amplicons ideally comes from a different species, for example, within that sample. And the colors represent the different index combinations that we have in these tubes. So what we do is we pool them in equimolar amounts. And in that way, we make them ready to load them into this sequencer. And here, the pooling sometimes can be tricky, so it's important to quantify our libraries accurately and to be precise when pipetting. So yeah, once we have our pool, we're going to sequence it. And the usual sequencer of choice is the Illumina MySeq machines, but we're also going to see throughout this workshop that there's going to be people talking about maybe different sequencers and different ways of doing things. So yeah, we load the library into the sequencer, and what we get out of the sequencer are millions of sequences that are sorted by their sample of origin. So we're going to end up with several computer files that are going to contain all of the sequences for a certain sample. And once we have that, what we do is we run those sequences through a bioinformatics pipeline. And we're going to compare those sequences to a reference database, and we're going to end up with the species composition of each of the samples. I'm not going to get into much more detail here because there's going to be people talking about bioinformatic analysis, and also after the workshop, some people have signed in for the DNA. So for the DNA metabarcoding data analysis course, where Neus will explain this a lot better than I ever could, and so I'm gonna leave it here. I just want to leave you guys with some notes, with some take-home points. So the first one would be design your experiment, especially design and select the best primers for your purposes. Avoid cross-contamination at all costs, because as I said, high-throughput sequencing technologies are so powerful that contamination is going to show. Also, design your bioinformatic analysis accordingly. So you need to select your reference database. You need to select the parameters that you're going to use for each of the scripts. That's really important as well, because results can vary a lot depending on the bioinformatic analysis that you perform. And this seems, I guess, I don't know, maybe, but this seems pretty straightforward. But it does require a lot of optimization once you get your hands on some samples. You want everything to be as good as possible. So you want to get the best DNA extractions. You want to get your PCRs to work, and you want to be able to analyze your sample in the most optimal way. So yeah, there's a lot of work behind each project of just in optimization issues. And so that's it on my part. Thank you all for listening, and I'll be happy to take questions.
Thank you, Anton, for your interesting speech. I think that is a very good way to begin this type of workshop. Do you have any questions? No questions. Okay, Anton.
Yeah, I think.
You said that when you mix the PCR products from different samples, it's quite important to quantify the amount. Because at this step, if you mix different amounts from different samples, I don't really get which.
Okay, what the problem is? Okay, yeah, we will also. I think we will have some people talking about that as well. I didn't get into much detail, but so sometimes it may not be that important, but if you're doing an experiment where you want to get the same number of sequences in each of your samples, you need to pool them accordingly. Because if you pool one sample more than the other, you're going to end up with a lot more sequences for that sample than for the other one, and that may bias your analysis. Or maybe not, but you can come out short of sequences for one sample and overkill it with the other sample. So.
But you could analyze at the bioinformatics step. You could
normalize the.
Resample the.
Yes.
The results.
Yes, you could. That can be one way of doing it. But say, for example, that you end up with too few sequences for one sample. That's a problem, and you may need to resequence, and that's more work and more sequencing and all that. So to start, I wanted to show that it's kind of important to make sure that you're pulling your samples correctly. But sometimes it's not possible, and sometimes you just have to deal with what you have. So you'll need to adapt your experiments accordingly. That's why I was talking so much about optimization at the end there, because you really need to amplify all of your samples so you can quantify them and pull them as equimolar as possible. Yeah.
I was wondering about the temperature in which you are storing your samples when you are transporting them, say, from the field to the lab. What's more important is like specific temperature, or it's more like keeping it constant, or?
I think okay. So I think it's important to keep them cool at least. So I think 4 degrees, because I understand that if you're sampling, there's not always the possibility of keeping your samples on dry ice, for example, which would be the best thing. But yeah, constant temperature is one of the points. Then if you can freeze them, that would be awesome. If not, just cool them, just to make sure that the DNA doesn't get degraded on the transport to the lab, and that it doesn't suffer variations of temperature.
Thank you for the explanation of the PCR steps. I understand that the adapters, they are technology specific, so
yes.
adapters you have to buy them. But,
well, not necessarily, but.
So you can actually order them from a company.
Yes.
In other words, are those sequences public?
Yes. Well, they're sort of public. No, I mean they are, and you can order them and just make your own oligos.
And what about those those indexes, primers, and the PCR primers? Of course, you have to design them. But the other two.
Yeah, those are also.
It's your design. You retrieve them from literature.
So okay, the
forward and reverse index.
Yeah, the forward and reverse index, you can design them yourself. These are just 8 base pairs, and so you can actually. There is some controversy about that, because there are some indexes that are known to work better than others. And in fact, some sequencing companies are telling you not to use certain indexes, but those are public. There are a lot of index sets published everywhere, and this sequencing primer Illumina has its own sequencing primers, but you can use your own custom primers if you need to for some reason. And the adapter is universal because it's going to be needed to attach to the flow cell.
That's why I thought it was technology specific.
Yes. So this amplicon construct is going to change depending on the platform you use. I've shown here the design for Illumina, but yeah, it's going to be a bit different for other platforms.
And second question
Yes.
is more curiosity. You mentioned the need of being accurate by pipetting. So I'm curious how you keep those measures in the lab. For instance, how often do you calibrate your pipettes?
Right. I think we've established our calibrations once a year. But yeah, I don't really know how much they can. I mean, when I say be cautious when pipetting, I meant that 2.5 is not 2.55 or 2.6. That you want to be precise when doing that.
And more about your hand.
Yeah, and also make sure that your pipettes are okay and that your material is working okay. But that goes for everything. So for the qubit where you quantify your samples, for everything, and you have to make sure that your material is working correctly. Yeah, that's a point I didn't make in this presentation, but that goes without saying that you need to have your material in optimal conditions. Yeah.
A question. In the first PCR,
Yep.
is it possible to use an internal standard?
Sorry, what?
Internal standard to put a sequence to quantify how well your PCR runs.
Yes. So you can do that. What people usually do is to use what is called a mock community. So that's a fake sample that you make in your lab from known quantities of DNA, and you add that as an extra sample to the experiment. And what you're doing there is you're running the very same PCR, so it's like a positive control, and then you see how much you can retrieve and see how well your PCRs are performing, and see how many taxa you're able to amplify, and if that correlates to your mock community that you have made.
But you don't have your inhibitors in that community.
That's true. Yes. Yes, but then if you, I mean, I guess you could do it. But if you add a sequence, some DNA to your sample, you have to make sure that you don't have that DNA in your sample, in your original sample. But yeah, that'd be a good thing to test. Yes. So I don't know if yes.
Since you have to stick to some taxonomic groups, isn't this an important bias in your results?
What do you mean?
I mean, you said that you have to stick to some taxonomic groups. Okay?
Yes.
Is this an important bias in your results?
Well,
you can only find what
you're looking for. Yes. Well, when I meant taxonomic groups, I meant but like the big taxonomic groups, so bacteria or fungi or eukaryotes, or but you can actually target different taxonomic groups in the same experiment. You just have to do it separately. So you may be interested in invertebrates and bacteria, or in amoeba and fungi, or whatever. So you can use different primers to amplify each of those barcodes, then pull them all together and get the sequences as if it was an extra sample. So you'd have to do the analysis twice. So there are not universal primers to recover all of the biodiversity that is in the sample, but only the ones you are actually targeting. So yes. Yep.
I have a question regarding the library pooling also.
Yes.
if you are interested to know the abundance of the species, to relate the number of sequences that you get during sequencing to the abundance of the samples, of each species in the samples, is there a conflict when you pull the library, when you do the pooling at the molar
Okay, so
concentrations? You know what I mean?
yes.
Does it change the original abundance of each species in the sample?
Okay, it shouldn't, because it should be proportional. So you're always adding the same amount of DNA to the pool, and that amount is expected to be representative of the original sample. But you make a good point there about the method being quantitative if you want to tell the abundance, and it's not entirely quantitative. So you need to design your experiment in a way where you add known mock communities and known concentrations, and you need to have some standards in order to calibrate the analysis, in order to make the method somewhat quantitative. Yes. I don't know if there's any more questions.
Any more questions?
Okay. Thank you very much.